Foundation Module 9 is Modular Integrated Undergraduate Curriculum Physiology. Dr. Sheen, CEO and Founder. Medical Superfast Simplified Physiology Video Lecture Notes. Guyton and Hall 14 edition, chapter number 51, lecture number 12, page number 641, 642. Learning objectives. Describe the rhodopsin-retinal visual cycle, or photochemistry of vision. Significance of visual cycle. The rhodopsin-retinal visual cycle, also known as the phototransduction cascade or visual cycle, is the biochemical process by which light is converted into electrical signals in the photoreceptors, rods, of the retina. This cycle involves the photopigment rhodopsin and its chromophore, 11-cis-retinal, and it is important for vision, especially in low-light conditions. Photochemistry of vision. The rods and cones contain chemicals that decompose on exposure to light, and they excite the nerve fibers leading from the eye. The light-sensitive chemical in the rods is called rhodopsin, the light-sensitive chemicals in the cones, called cone pigments or color pigments. Rhodopsin and its decomposition by light energy. The outer segment of the rod that projects into the pigment layer of the retina has a concentration of about 40% of the light-sensitive pigment called rhodopsin, or visual purple. This substance, rhodopsin, is a combination of the protein scotopsin and the carotenoid pigment retinal, also called retinine. Retinal is a particular type called 11-cis-retinal. This cis form of retinal is important because only this form can bind with scotopsin to synthesize rhodopsin. When light energy is absorbed by rhodopsin, the rhodopsin begins to decompose within a very small fraction of a second. The cause of this is photoactivation of electrons in the retinal portion of the rhodopsin, which leads to instantaneous change of the cis form of retinal into an all-trans form. Because the three-dimensional orientation of the reactive sites of the all-trans retinal no longer fits with the orientation of the reactive sites on the protein scotopsin, the all-trans retinal begins to pull away from the scotopsin. The immediate product is bathyr-rhodopsin, which is a partially split combination of the all-trans retinal and scotopsin. Bathyr-rhodopsin is extremely unstable and decays in nanoseconds to lumirhodopsin. This then decays in microseconds to metarrhodopsin I, then in about a millisecond to metarrhodopsin II, and finally, much more slowly, in seconds, into the completely split products scotopsin and all-trans retinal. It is the metarrhodopsin II, also called activated rhodopsin, that excites electrical changes in the rods, and the rods then transmit the visual image into the central nervous system in the form of optic nerve action potential. Reformation of rhodopsin. The first stage in reformation of rhodopsin is to reconvert the all-trans retinal into 11-cis-retinal. Once the 11-cis-retinal is formed, it automatically recombines with the scotopsin to reform rhodopsin, which then remains stable until its decomposition is again triggered by absorption of light energy. This process requires metabolic energy and is catalyzed by the enzyme retinal isomerase. Significance of the visual cycle. The visual cycle allows the photoreceptors to continuously respond to varying light conditions by regenerating the light-sensitive pigment, thereby enabling vision in both dim and bright environments. Since retinal is derived from vitamin A, a deficiency in vitamin A can lead to impaired vision, particularly night blindness, due to the inadequate regeneration of 11-cis-retinal.